Welcome back. This is chapter 7. This is one of my absolute favorite chapters because we're going to focus on controlling microbial growth. And depending on how your mind is working right now, whether or not you're thinking about it from the aspect of a microbe or an aspect from a human, you would approach this in two totally different ways. So we're going to be taking a look at microbial growth and seeing some of the things that we do or maybe we don't do, some of the things that you may not be thinking about on a day-to-day basis that does play a pretty big role in how microbes are able to grow and survive. When you look at controlling microbial growth, to be honest with you, we're not going to be septic in our homes or in our cars, at our work, wherever we're out to eat, something like that, because working in a septic environment means that there's absolutely no microbial contamination. The best that we can offer as far as lab purposes, as far as when you're disinfecting areas and things like that, is to really work for asepsis, which asepsis is the absence of any significant type of contamination. So I'm not looking at something that is going to be completely without microbial growth. What I'm looking for is something that at least is not significantly contaminated. Of course, in areas something like surgery, where we're going to be trying to be as sterile as we possibly can, we definitely want to prevent microbes from being there so that they don't get into wounds or cuts or any openings that we may have in the surgical field. Some terminology that you guys need to get used to thinking about. Number one is sterilization. When you go into a lab, and we're going to talk about the autoclave and talk about what that is, one of the things that we have to do, though, is we have to be able to sterilize the tools that we're going to use in order to grow these microbes out, like our plates, right? We talked about slants last time, but now we're going to be looking at agar plates. And so when I sterilize those, that means that for that moment in time, I'm going to remove every single microbial life that happens to be there. In your homes, you do things that are called disinfecting. Disinfection, by definition, is the removal of pathogens. Now you think about what that means. Number one, did I say killing? No. I said removal of pathogens, which means that you're simply taking it from one surface and you're putting it on another surface. So for those of you who use something like paper towels, washcloths, hopefully you're not doing sponges, anything that you're going to be cleaning your surface with, though, you're really only taking the pathogens off of your countertops, for example, and just putting them upon whatever you're cleaning with, whether that be that paper towel or that washcloth or something like that. We also work to de-germ an area. Usually when you're cleaning, you're very concentrated, like maybe you're going to clean the bathrooms or maybe you're going to clean the kitchen, but you're very concentrated on an area that is a limited area that you're going to be working with. We worry a lot of times about sanitation. I can tell you right now, if you are not concerned about sanitation and you're going into restaurants and you're not looking at what was their sanitary score type of thing. I've walked into a restaurant, well, just a couple of weekends ago. The girls wanted to eat Chinese food after their competition in gymnastics. And so we pulled into a local restaurant there and started to open the door. And on the door, it said 74, I think is what it said on their sheet for their sanitization from the health department. And I said, oh, no, 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 no. We are not eating here. And we turned around and we left. Some people are like, well, I don't want to do that because it's rude. I would much rather be rude per se than to get sick with an E. coli infection or some sort of salmonella or staphylococcal food poisoning. So I'll take my chances with saying, you know what, excuse us, I think we're just going to go somewhere else. So be careful with where you're eating and things that you're doing around your house, because a lot of times you don't think about things that necessarily are just spreading germs from one area to another. I know my mother is bad about getting a washcloth out, wiping down countertops, and then laying it across the sink. All you've really done is move the microbes from the countertops over to the washcloths, and now you have a new environment for those microbes to actually grow upon. We talked about the fact that when bacteria grow, they grow logarithmically, which means that they grow extremely fast. They grow 10 times the amount. But we also talked about the fact that when they die, they also die logarithmically. So once microbes start to die out, they do die out at a very, very fast and constant rate. When you choose an antimicrobial treatment for your house, and for lots of people that's different things. Some people utilize Mr. Clean or Lysol, or some people use bleach, or some people use vinegar. When you choose a treatment though, there's some things that you really have to be cognitive about if you're going to be effective in the way that you use those treatments. Number one, it does matter how many microbes that you have. If you are cleaning your homes once every week versus every single day, or once every month versus every other day, the amount of microbes that you are trying to get rid of at that time is significantly different. I know for a fact some people choose rooms, like today I'll choose the living room to clean, and tomorrow I'll choose the bathroom to clean, and tomorrow. And so what they do is every single week they cycle through that. But what that means is that those rooms, like the bathroom or the kitchen or the living room, haven't been cleaned in 1 week's time. We already talked about the fact that microbes multiply in 5 hours. If you have 100 cells, you're now already looking at over 1.7 million cells. And so imagine where you're at now if you're only doing this for every other week. The number of microbes can get out of hand very, very quickly. It also does depend on the environment that you're in. What's the temperature? What's the organic matter? Are there any biofilms that have been built up? For example, in the bathrooms, right, where we know there's extra microbes that are being simply growing just because of the nature of what's there. Big one, huge one, is time of exposure. I see people cleaning, and I watch people do it in lab. I watch people do it in their homes. They spray down their countertops with whatever they choose, and then they wipe it right up. And they're very happy while they're doing so. Take a look on the back of your cleaning bottle and look what the directions say. Not very many people use the directions on cleaning bottles because who doesn't know how to clean, right? But the thing about that is that it does have directions and should probably say to leave exposed to the surface for 10 minutes at least. That would be a minimum. Because in order for a treatment to be actually effective, it has to sit on those floors or on those countertops or on those sinks for at least the appropriate amount of time to be considered antimicrobial. If you're spraying and wiping down, you're essentially not doing anything antimicrobial. All you're doing is removing those pathogens to another surface and giving them a new platform to grow upon. You check your cleaning supplies at home and see just how much time of exposure you're supposed to have. Another thing I find very interesting is that people buy stuff and they have no idea what the active ingredients are. Some people buy things suspecting that they would be good as far as killing microbes when really they have absolutely zero antimicrobial properties. Great for dirt, great for dust, great for grime, but does nothing for microbes. Make sure you're checking what you're using at home. We also see microbial characteristics. The type of microbes that you have in your house make a difference. If you are being exposed to some of those pathogenic microbes that are really serious versus just some of the everyday common types of households, that's a difference. Think about the stomach bug, right? Now, I know that's viral and we're not talking about viruses right now, but just think about the stomach virus. Think about how you have to clean differently. Doesn't it seem like you have to get in every nook and crevice and cranny in order to clean that particular bathroom or that particular kitchen to make sure that you get rid of all of the microbes that are present? That's because those microbial characteristics are very different from some of the characteristics that we see on a day-to-day basis. When we look at how to pick a chemical to treat microbial growth, we know in a lab that there are certain parts of a microbe that if we target and if we damage, those particular damaging factors will ultimately lead to the death of the microbe. So if I can hit the plasma membrane, hit the cell wall, damage their proteins, damage their DNA, all of those are actions that we take in order to specifically target microbes and then cause damage to those areas, which would then ultimately lead to death. When we look at methods that we can use, and you can do this on a day-to-day basis, some of them aren't quite realistic to do on a day-to-day basis, but when you look at methods that you can do to control the amount of microbes, an easy one that we've known for a very long time is heat. High amounts of heat, we know, denature enzymes. They break those three-dimensional shapes into a linear shape, and you guys know that those linear shapes are inactive. We know that microbes can live up to a certain point. So there is a certain time when microbes can be surviving even if you turn up the heat just a little bit. But over time and after a while, if you keep adding heat to those microbes, eventually they do die out. One of the things that we find that in the lab that we do is we combine heat with steam and pressure. So steam, the heat would be in the form of steam, and then the pressure that we utilize is coming in order to make things sterile. This that you're seeing on your screen here is an autoclave. We have an autoclave in the lab that we utilize to clean our glassware. We utilize to kill microbes with this because what it does is it combines this steam, and under high amounts of pressure, it would provide us a way that we could be sterile for that particular time. So right now you're looking at a beaker of something inside of the autoclave, and we're adding 15 psi's, which is pounds per square inch, at 121 degrees Celsius, and we run the autoclave for 15 minutes. And what we find is that when we take that beaker out of the autoclave, whatever is inside there is completely sterile. Now, of course, if I open it up and expose it to the air, it's now become contaminated. We use steam all the time in medical practices. I personally at home use steam. I love my floors to be steam, my countertops to be steam. It adds that really high amount of heat in steam to contact the surface and does a really great job in sterilizing different surfaces. We can also do pasteurization, which is not the same as sterilization. We do know that organisms survive pasteurization, but the thing about them is that they're not potentially giving us any type of issues as far as disease or spoiling food. So we can run these different treatments, and they're all equivalent. You can do 63 degrees Celsius for 30 minutes, or you can do 72 degrees Celsius for 15 seconds, or you can do the ultra high, which is 140 degrees Celsius for anywhere less than a second. But the thing about this is it's not going to wipe out all microorganisms. It's going to make whatever you are trying to do, let's say milk, for example, because you know you pasteurize milk. It's going to reduce the organisms that are going to cause harm, disease, and spoilage to the milk, but that doesn't mean that that milk is sterile. The milk that you have still has cultured microbes that can be grown from it, even if it's pasteurized. One thing that we do in the lab also that does do a very good job in sterilizing things is to simply put it in an oven. We'll run glassware once you clean it out. We have an oven in the back of the lab that runs at 170 degrees Celsius that we put them in there for about 2 hours and bake them, and that actually is equivalent to being sterile. We know that we can filter out microbes. We talked about filtration. We know that we can add a lot of pressure. We know that we can change their water content. We know that we can put them in an environment that's either hypotonic or hypertonic that can help control these microbial growths. We know that high amounts of X-rays or gamma rays, those do actually control microbial growth by basically damaging the DNA. We also know that microwaves are not potentially one that would be utilized to control microbial growth. It's interesting that people think that microbes cannot grow in a microwave. They absolutely can. Now, let me ask you this. Can microwaves boil water? Well, the answer is yes. But how long would you have to set a bowl of water in there? It's usually about 3 minutes, right? 2.5, something like that. And you sit it in there and you get it boiling, and then you would have to boil it for at least 20 minutes. A lot of people don't realize how long microbes can actually live inside of boiling water. Staphylococcus, that makes you sick with that kind of 24-hour stomach bug, it's a food poisoning. And it can survive to up to 30 minutes in boiling water. You think about that. How long do you boil a hard-boiled egg? 10 minutes? 15 at the most? Certainly not 30. And so when you think about controlling these microbes this way, it's just not feasible to think of a microwave as being one of the ways to kill microbes. There are chemical methods that we mostly choose. I mean, most of the time people aren't going to go in and crank up their thermostat in order to control the microbes in their home, but they definitely will choose a chemical to do their floors or do their kitchen countertops, to do their sinks or their bathtubs. Of course, there's no single disinfectant that's going to kill everything. You have to be very smart about the chemicals that you use. I know a lot of people who do the kind of all-natural, go green types of things, where they're using vinegar or they're using baking sodas, which is fine as long as you are up to par on what exactly you need to be doing in order to make sure that those things are antimicrobial. When you look at the effectiveness of a chemical, you have to know the concentration of the disinfectant that you're using. I don't know how many times I've seen people open up a thing of Lysol or open up a thing of whatever it may be, Kaboom or whatever, and just dump it into a bucket or dump it into their sink, and they don't actually have any idea the concentration that's actually needed in order to kill these microbes. Another one is the organic types of matter that it's made of. Another is the pH. You can't utilize something that's neutral and try to kill microorganisms. You know that microbes don't like too acidic and they don't like too basic. So we play on that pH in order to make sure that microbes are actually being killed. We also know time of exposure is a big one. You have to leave that chemical on there long enough for the microbe to actually become affected. In a lab, one of the things that we want to know is whether or not a chemical is going to be effective against particular types of microbes. If you look on the back of, I keep coming back to Lysol just because it's kind of a generalized name that we can use, but if you buy Lysol and you look on the back of it and it says 99.9% effective against Staphylococcus aureus. Well, how do they know that? Right? You can't just say that. You can't just claim that. There has to be a test in order to prove that. And there is. We utilize all the time in labs chemicals to test whether or not they're effective against certain microbes. For example, you can take a metal ring and dip it down into a known bacteria. So let's say E. coli. You can dip that down into E. coli and let the E. coli dry on that ring. Then you take that culture and you put it down in your Lysol and you let it sit at room temperature for 10 minutes. Now, room temperature is very important because that's where you're going to be utilizing these chemicals, right? You're going to be utilizing them in your homes, in your office spaces, where it's going to be nice and room temperature. So I don't want it to be too cold or too hot when I'm looking at this control center, because I need it in areas where you're going to actually be using it. And so we let it sit for 10 minutes, and then you bring that ring up and you put it on a culture media. Well, if something grows, that tells me that that Lysol is not effective against that particular microbe. But guess what? If nothing grows, well, if nothing grows, then I can put that on the back of my labeling that says, hey, I'm 99.9% effective against E. coli. And so they test all kinds of different chemicals and all kinds of different microbes against the concentrations of chemicals and the use of chemicals so that we can know whether or not they're going to be effective. In our purposes, we would use what is called the disc diffusion method. Now, normally I don't have these little discs that are full of chemicals, but I do have them that are full of antibiotics, and it's the same concept. We're essentially looking for, do these chemicals treat certain known bacteria? And what you look for is a clearing around the disc. It's called the zone of inhibition. The larger the zone of inhibition, the more effective that treatment actually is. So I want to take a look at that first one, which is the Staphylococcus aureus. And when you look on there, you can see that all of them are actually effective because they all have a zone of inhibition. But there's definitely two that seem to be a little more effective just because that clearing around the disc, that zone of inhibition, is a little bit larger. Take a look at the center one because I'm going to pause after this and take a break. But I want you to think about a couple of things as I leave you here. So this is an agar plate that we have swabbed with E. coli. And E. coli, as you know, is found in the colon, right? That's Escherichia coli. Take a look at those four things. I have chlorine, I have O-phenylphenyl, I have quaternary ammonia, and I have hexachlorophene. And I'm going to ask you a couple of questions. Number one, do all four of these chemicals treat E. coli? No, right? Which one does not treat E. coli? The hexachlorophene, right? And I know that because there's no zone of inhibition. When you look at the microbial growth, it is growing all the way up and around that hexachlorophene. The next question I want to ask you is, which is the most effective? Well, chlorine is, right? You see, chlorine has the largest zone of inhibition around those microbes are growing, and you can see that zone is rather large. O-phenylphenyl and quaternary ammonia could be used, but if I had my choice, I would pick chlorine. So let's talk about a couple of things real fast. Number one, E. coli is found a lot at high concentration, obviously, in the bathroom, right? Because if it's in the colon, it is a high concentration in fecal matter. So chlorine treats it. O-phenylphenyl treats it. Quaternary ammonia would kill it, but hexachlorophene does not. Chlorine is things like your bleach contents, right? You've got chlorine tablets, you've got bleach tablets. You can mix those up together and kind of use as a cleaning solution. O-phenylphenyl is Lysol. We actually have a chemical compound that tweaks the natural phenyl that is found in nature, and that's what O-phenylphenyl is. Quaternary ammonias are an ammonia type. You guys have heard of ammonia, right? It's an ammonia type of chemical, so that's more of a basic type of chemical. And then there's hexachlorophene. Hexachlorophene is antibacterial soap. Now you think about when you go to the bathroom, what do you do as soon as you use the restroom before you leave? You wash your hands, right? You wash your hands, but yet I just told you that hexachlorophene does nothing against E. coli. You think about that. We're going to do a hand washing lab together. We're going to talk about some of the very, very interesting things that come with just how you actually wash your hands and what you're doing when you do wash your hands. So hexachlorophene, though, if we look at the next slide, I believe, yeah, there's phenylphenol. O-phenylphenol is Lysol. Lysol actually works to break down the plasma membrane. And then hexachlorophene is your antibacterial soap. Also works to break down the plasma membrane, but it's probably not as effective as you actually think. So I'm going to leave you right there. I'm going to pause for a bit, and we will pick up with different types of disinfectants that maybe you use sometimes in your homes, and we will come back and talk about that soon. Okay? Ciao, guys.